Good afternoon and welcome to the Adelaide Biomed City Minireview webinar series. My name's Andrew Zannettino and I'll be your chair this afternoon. Before I introduce our first speaker, I'd like to acknowledge the Kaurna people, the original custodians on the land on which we meet here in Adelaide. And I pay my respects to elders past, present and emerging. I'd like to also remind you that you're able to ask questions at the end of the presentation from our speakers, and you can use the Q&A section under the Zoom window, and that'll allow me to relate the questions to our speaker. This afternoon, we're going to be hearing from two University of Adelaide researchers and who will discuss their research related to brain development and neurodevelopmental disorders. Our first speaker this afternoon is Dr. Lachlan Jolly. Lachlan received his PhD in biochemistry at the University of Adelaide in 2010. He undertook postdoctoral research training with neurogenetics research program headed by Professor Joseph Gex. And in 2016, Lachlan was awarded a prestigious Australian Research Council DECRA Fellowship and is now currently supported by a first 1,000 days fellowship. In 2021, he was appointed to the group head in the School of Biomedicine at the University of Adelaide, where he leads the neurobiology research group. Lachlan's uses brain in a dish models to investigate the genetic and cellular mechanisms of embryonic and childhood brain development. And the title of Lachlan's presentation this afternoon is The Mechanisms of Brain Development. Welcome, Lachlan.
Thanks, Andrew. A lot of my research is kind of around solving these problems of how you actually study the mechanisms of brain development, where it's very difficult to actually get your hands on the material of a developing human brain. And I study the mechanisms of brain development, as Andrew mentioned, with a vision that would like to provide children with neurodevelopmental disorders, and these disorders usually arise because of aberrant brain development, how we can provide these children with options tailored to their underlying genetic causes, because in Western countries like Australia and all over the world, most neurodevelopmental disorders do have a genetic cause underneath them. So to achieve this, we're really looking at a somewhat generic pipeline to precision medicine, where we need to obviously identify the genes involved right at the beginning of this pipeline. And I've been involved with this mainly through collaboration with Joseph Gex over the last 10 or so years, and providing diagnosis to families has many benefits. I'm sure Claire will tell more about this in a minute. And it's really about assessing whether a particular genetic change in a child, and we call these changes variants, are actually pathogenic or just part of natural variation. And we get these variants which look quite suspicious, for example, those that might be sitting in a known disease-causing gene. And a lot of the studies I have contributed to is really about providing additional evidence to try and move this needle towards a pathogenic outcome where we could clinically report a finding for the family. I've been working with one particular gene which I discovered in 2013 involved with neurodevelopmental disorders called USP9X, and over the last 10 years or so we've been generating predictive tools that's to help assess variants just based on DNA sequence alone. We've been accumulating the clinical data of these patients. It's a very rare disorder. There's only probably a couple of 100 patients known. So actually accumulating this data really helps the clinicians identify what children with changes in this gene present like. This sort of data is captured for diagnostic resources, and we also actually brought the families together as a community, which is quite nice because, as I mentioned, it's a rare disorder. So actually communicating amongst one another is actually a good way to help provide treatment for these children at this stage. Another thing we've been interested in kind of lately is doing functional studies. So that is to say, where we have a variant, we really like evidence to show that that variant is somehow detrimental to the gene and therefore the protein's function. And for this, we like to use cell lines from the patient. These are blood-derived cell lines or can be skin-derived cell lines. And that's quite useful resources for many genes. But actually, one problem that we're trying to solve at the moment is that of the around about 5,000 Mendelian disease genes, about a third of them actually aren't expressed in blood or skin. So how do you do functional studies on these types of genes? About 500 of those genes are actually neurodevelopmental disorder genes. So we've been using a technology based on deactivated Cas9, which you're probably aware of as a gene editing tool. So we inactivate it and basically use it as a courier to try and wake up these sleeping disease genes actually in patient cells. So we load up this Cas9 molecule with transcriptional activators and send it to the promoter of choice, and it turns on transcription. So these are a number of disease genes involved in epilepsy and neurodevelopmental disorders, for which now we can actually turn on the gene in the patient fibroblasts, which means for the first time we can actually get access to the RNA to begin to do the functional assessment on whether a particular gene variant is actually altering the function of the gene. Otherwise, we would have to go to a brain biopsy or perhaps more sophisticated models. So this looks like quite a nice way forward, and we're pretty excited about this. So providing diagnosis is, of course, just the beginning of the journey. As I mentioned, the long-term idea of the lab is to really get our hands on some precision therapies. And I'd like to just walk you through some of the ways we look at this middle bit, understanding mechanisms of pathology. As I mentioned, it's difficult to get your hands on the actual material. So we basically use, as Andrew said at the start, brain in a dish models. We can generate brain cells from stem cell models, from mouse and from human. We also use mouse models themselves, mainly through collaboration. We do neuronal cell models where we can take neurons out of the developing mouse embryo and culture them and manipulate them. And more recently, we've been actually using induced pluripotent stem cell technologies to reprogram patient skin cells actually into stem cells and then on the way to neurons, so we can actually get our hands on patient neurons in the dish. The good thing about all of these models is that they not only help us assess whether gene variants, and these two genes in particular, are causing disease, but actually gives us indication on what the effect of those mutations is actually doing. For example, here with in this couple of publications on this gene USP9X, we identify mutations, but we also identify that they actually disrupt neuronal cell migration and growth, or TGF-beta signaling. And likewise with this other gene that I'm really interested in, we understand that mutations actually alter the way that the neural stem cells of the growing brain behave, and that it also regulates a large number of gene networks involved with neurodevelopmental disorders. And so towards getting understanding of these mechanisms is really helping us, I guess, on our way to treatments. But I guess it's foolish to maybe consider that we'll be able to get a precision treatment for every gene involved in neurodevelopmental disorders, because there's greater than 2,000 of them. What we'd like to do, though, is work out where these genes somehow converge. They all converge in the end to control brain development to at some point. So if we can kind of target points down towards the lower end here, we may have one treatment that might be suitable for the treatment of many types of genetic causes of neurodevelopmental disorders. And studying these rare cases of USP9X and UPF3B has actually led us to some of these points of convergence. UPF3B, in fact, is involved in a process called nonsense mediated mRNA decay, and this is a major focus of the laboratory studying this process. This particular pathway degrades all mRNAs that harbour a type of mutation called a nonsense mutation. And that's actually ends up being a modifier of around about 30% of all genetic diseases. So it's a big player in genetic disease in general. USP9X, this gene ended up being one that controls the stability of other proteins through the ubiquitin proteasome system. And the altering of protein dosage, well, actually the loss of protein dosage, known as haploinsufficiency, is actually also relevant to around about 3,000 genetic diseases. So these two pathways are ones that we're trying to maybe target therapeutically. For nonsense mediated mRNA decay pathway, we've been generating sort of live cell biosensors that enable us to do high throughput content screening of drugs We're working on identifying biomarkers to validate these drugs, and also to triage different patients into who might be best responsive to these drugs. Here's samples of 100 different healthy individuals, and the ranking of them basically tells us whether they're likely to respond to NMD inhibition or not. With the protein stability side of things, we're really interested in working out ways to manipulate the ubiquitin proteasome system to increase dosage of proteins that are causing disease through loss of dosage. So that's essentially repurposing drugs from the cancer field. They've long recognised the importance of this protein, or the utility of it as treatment. So we either look at E3 inhibitors or activating the DUBs to prevent the degradation of proteins. So this project sort of involves a lot of protein-protein interactions and proteomics. So with that, I hope I've given you a bit of a snapshot of the techniques and the processes in the lab we're interested in. A lot of the work's done in conjunction with Joseph's neurogenetics group, and I won't go into too much of the acknowledgements, other than to thank the patients and families whose contribution to the research is invaluable. So thank you.
Thank you, Lachlan, for a fantastic presentation. As I indicated earlier in the introduction, please feel free to ask questions of our speakers today using the Q&A function. While waiting for some questions, I might start, if I could, Lachlan, just in respect to the fact that a couple of the genes that you've identified have neurodevelopmental function converge around areas which are clearly key biological functions that may, in fact, if inhibited or impacted by therapy, could have implications for other systems within the body. Are there anything? Is there a very specific part of those pathways that you're starting to look at repurposing existing drugs, or looking at ways in which you can interrogate that more specifically?
Yeah, I guess there's a couple of points there. One is that I guess it's the window of opportunity to intervene is interesting, because these pathways and molecules that we're looking at are very important for development of the brain. But for example, the nonsense mediated mRNA decay pathway seems a little bit dispensable in adulthood. So if you can actually manipulate it without causing too much problems to the homeostasis of an adult mouse. So certainly the window of opportunity there is to manipulate after development, where it seems to be really important. And I guess the other aspect of it is working out the precision. How do we target these molecules in the right cells at the right time? That's probably a little bit outside of my expertise, but delivery techniques into brain and gene therapies or small molecule therapies that are brain targeted are certainly an active field of research that hopefully we'll be able to tap into and collaborate within as we find the key molecules which are actually having biological effects. So yeah, it's a big field, and one that's quite daunting to step into, but the development of therapies, through collaboration with research and biotech, I guess, is where we're headed in the longer term.
Just as a way of following up, in terms of the frequency of these disorders, I mean, clearly you indicated that they are not incredibly frequent, and in fact you are reliant on accessing tissues from patients in order to start to investigate or get an understanding of the mechanisms that's underlying their particular disease. Clearly you're seeing a high degree of variation between individuals, and particularly in pathways or parts of pathways that lead or may converge on similar pathways, resulting in the phenotype. How in most, can you give me some sense of the variability that you're observing amongst the genetic predisposition, or can you give me a sense of that? Because I don't have any feel for that at all, the recurrence of these particular mutations or in a particular phenotype. Yeah,
they are quite rare. Each one of the disorders I've worked on, there's probably only a few hundred people worldwide that are known, and I guess if you can probably double or triple that number if if individuals in countries that don't have access to genome sequencing were included as well. So altogether, yes, they're quite rare, but rare disorders as a collection are actually quite frequent. I think, there are 2,000 genes involved with neurodevelopmental disorders, and around about 95%, I would say, of people with a neurodevelopmental disorder will have a mutation in a rare gene. The most frequent cause of intellectual disability, for example, is Fragile X, and that still has a very low prevalence in the population. So yes, we really do try to work on those points of convergence. So for example, that UPF3B gene, where we know that causes compromisation of this nonsense mediated mRNA decay pathway. So we first discovered that gene, I think, in 2008. But since then, all the other genes in the pathway have since been implicated in an overlapping disorder. Right. So the one pathway can be disrupted by various different types of genetic insults. And we see that quite often. We have the ciliopathies, for example. So these are all genes involved with formation of a cell structure called the cilia, and they all have a very overlapping sort of manifestation in disease. That's microcephaly. So yeah, finding those points of convergence, grouping the genes together instead of as single genes, but in pathways, is the way that we can, I guess, get our head around how best to design therapeutics that are not going to be just for one gene, but across multiple different genes.
That's fantastic. Look, thank you, Lachie, for a fantastic presentation and answering my very basic questions. But thanks.
They're excellent. Thank you.
Look, thank you. Look, that leads me to welcome our second speaker for this afternoon, and that is Dr. Clare Van Eyk. Clare completed her PhD in 2010 with Professor Rob Richards at the University of Adelaide, where she investigated the pathogenic mechanisms in the dominant expanded repeat disorders using a fly model. These dominant expanded repeat disorders include diseases like myotonic dystrophy and Huntington's disease and common forms of dominantly inherited ataxia. In 2013, Clare took on a postdoctoral position with the Adelaide Cerebral Palsy group, where she now works across neurogenetics and cerebral palsy research groups with Professor Joseph Gex and Emeritus Professor Alistair McLennan. Clare currently holds the Hospital Research Foundation Fellow and co-leads the Australian Cerebral Palsy Biobank. Clare's research focused on defining the genetic causes of cerebral palsy and related neurodevelopmental disorders by applying genomics and multiomics. The title of Clare's presentation this afternoon is The Genetics of Cerebral Palsy, and please welcome Clare. Thanks, Clare.
Thank you, Andrew. Just share my screen. I apologise in advance for my terrible voice. Okay, so today I'm speaking on behalf of the Adelaide Cerebral Palsy Research Group, and we sit in the Adelaide Health and Medical Sciences building on North Terrace. And our group really has key capabilities in gene discovery and characterisation of novel genetic disorders that fall under this umbrella of cerebral palsy. And we are applying multiomics to these disorders to discover also unusual causes and the mechanisms underlying them. And we do a little bit of animal and cell model work as well for specific genes that are of interest to our group. Our main collaborations are with the Neurogenetics Research Group, led by Professor Joseph Gex, who you can see in the photo here, and also with international groups through the International Cerebral Palsy Genomics Consortium, which we were involved in setting up back in 2017. We've also recently, with Zannettino, been involved with Panel Up Australia in setting up the first gene panel for cerebral palsy for diagnostic use. And I also have a collaboration with Twins Research Australia, which I'll briefly mention a little later on. So cerebral palsy is the most common physical disability in children, but it's not really a clear clinical diagnosis. It's more of a descriptor, and it covers a whole range of disorders, which broadly are permanent disorders of movement and posture, and they result from damage or abnormality of the developing brain, and that by nature they're defined as being non-progressive. So it's been suggested we should more correctly call them the cerebral palsy spectrum disorder because they are diverse both in the kind of damage that you see in the brain, in the pattern of the disability, and also in comorbidities that go along with them. There are a number of known risk factors for cerebral palsy, including pregnancy factors like prematurity, low birth weight, also infection in utero, and placental pathologies are known to be associated with cerebral palsy. But what I should point out here is that despite the traditional kind of perception, less than 10% of cerebral palsy can be objectively shown to be due to acute perinatal asphyxia. So this is asphyxia at birth. So these risk factors that have been associated with CP suggest that it's most likely due to long-standing intrauterine pathology in the majority of cases. The Adelaide CP research group have really pioneered genetic investigations for cerebral palsy since about 2014. And since that time, there's been a really rapid increase in the number of other groups publishing on this area. And in aggregate, there are now about 3,000 individuals with genetic findings published in the literature. And the diagnostic rate sits at around 30%, although there is a broad range depending on how the cohort is selected. There are more than 300 genes now associated with cerebral palsy, so it really is quite diverse, and a large proportion of them overlap with other neurodevelopmental disorders, including epilepsy, intellectual disability, and autism, which are also very frequently comorbid with cerebral palsy. So what we see is that you can frequently have one gene associated with multiple different kind of neurodevelopmental outcomes, and in many kids they actually have more than one of these disorders. The key resource of the Adelaide CP research group is our Cerebral Palsy Biobank. So over the last kind of 10 years, we've been recruiting to this biobank, and it's a repository of clinical data, medical imaging, DNA, and patient cell lines. And we now have more than 500 individuals with cerebral palsy in our biobank. And they roughly approximate what you would find in an Australian cerebral palsy register in terms of the type of cerebral palsy, sex, and comorbidities in the cohort. And over that kind of 10-year period, we've also accumulated a lot of multiomics data for this cohort. So we have exomes and genomes for the majority of our biobank now. We also have epigenetic data for a proportion of them. And also we've been doing a lot of transcriptome sequencing over the last couple of years. So what are we doing with this multiomics data? Well, the first thing that we can do using multiomics is to uncover cryptic genetic variations, so genetic variation that's not immediately obvious just from looking at a genome. And this is just one example of how this is useful to us. So in this example, we performed RNA sequencing and looked at outlier expression across the cohort. And we identified this one individual, patient 955, who was an extreme outlier for expression of this EIF2AK2 gene. You can see that's outlier expression compared to both all the other CP cases and all of the control sequence. When we went back and looked at the genome of this individual, we found that they actually have a rare homozygous genetic variant that's sitting just upstream of the transcriptional start site of this gene, and it falls within a region that looks like it's probably involved in gene regulation. It's reasonably highly conserved, and it actually looks like it probably affects this transcription factor binding site. This is a known disease gene, so EIF2AK2 is involved in a leukoencephalopathy disorder, so very likely this is the cause of this child's disability. So the other thing we're doing with this multiomics data is to look at convergent mechanisms. So I've told you all about how heterogeneous cerebral palsy is, but what we found when we looked at a large cohort by RNA sequencing is that there seems to be a gene expression signature, suggesting that there might be a convergent mechanistic pathway. So here you can see a heat map, cerebral palsy cases in green and controls in blue. And by looking at the top differentially expressed genes in the RNA sequencing data, we can fairly nicely cluster CP cases away from controls. And we were able to confirm this in independent samples by real-time PCR. And also we looked at gene networks in this data, and we were able to demonstrate that there is a network of genes which are co-expressed and enriched for BDNF signaling, which appear to be dysregulated in cerebral palsy. What's not clear at the moment is whether this gene signature is a cause of cerebral palsy or a consequence of the pathology in these children. So now I'm just going to talk about a couple of projects that are a bit newer to our group. And the first is one that Dr. Mark Corbett is running. And this is to look at common genetic variation in cerebral palsy. So everything I've told you about so far is really these high penetrant, very rare genetic findings that explain about 25 to 30% of cerebral palsy. And we have a few examples of intermediate kind of variants that are low frequency, but don't always cause disease. But what we're really interested in looking at now is these more common variants that might increase your risk of cerebral palsy, but on their own are not enough. So there's been one genome-wide association study published for cerebral palsy so far. It's not a massive cohort, and they did look only at spastic hemiplegia, but they found one SNP with genome-wide significance in this particular study. So what Mark's trying to do is collect a larger cohort and to look more broadly across cerebral palsy to try and identify genetic markers that might be associated with risk. What we can also do using this data is to develop a polygenic risk score based on summary data, not only for cerebral palsy, but other related traits such as prematurity and neurodevelopmental comorbidities. And the aim here is really not only to look at risk prediction for cerebral palsy, but to perhaps stratify this cohort into different etiologies based on the underlying genetic profile. So the final little project that I just wanted to mention is my Hospital Research Foundation funded project. And this is a twin study that we are still recruiting for, where we're doing multiomics on monozygotic twins who are clinically discordant for neurodevelopmental disorders, including cerebral palsy, intellectual disability, epilepsy, and autism. So we're doing deep whole exome sequencing on the twin pair and also exomes on their parents. And we're also doing RNA sequencing and epigenetic arrays on these twins. And the idea here is to try and uncover the mechanisms driving this clinical discordance. And what we can see so far is there are likely three separate kind of categories amongst these twins. The first are those who actually share a likely pathogenic variant, but they have differences in the way that they express that variant, likely due to epigenetic mosaicism. The second are the twins who have actually discordant genetic variation. So either one twin has a variant and the other does not. Or in some cases, they can actually both carry the genetic variant in a mosaic way, but one has a different variant allele frequency to the other. And in the third example, we can see quite extreme environmental differences from the pregnancy history of these twins. And that can either be on the background of a shared genetic variant or no obvious causative variant in the twins at all. So I just want to acknowledge everyone who has been involved in this work, particularly the Cerebral Palsy Research Group and everyone who has funded us. Thank you.
Thanks so much, Claire. That fascinating presentation. Look, thank you. Really, what caught my interest was some of the variations in the collagen variants that you've identified. Presumably, these collagen variants lead to intraventricular hemorrhage or bleeding, and I guess that's very much in the developmental stages of the birth of the prenatal circumstance. Then that leads to issues around the cerebral palsy. Is that how it's sort of? Yeah,
that's right. So they can also be associated with later intraventricular hemorrhage. Right. But there certainly are reports of them being involved in stroke in utero. And we have found a fair number of them actually in our cerebral palsy cases now. The trick with them is that they're not completely penetrant. So often they're inherited from a parent. And that parent obviously is still at risk of having a stroke later on.
Right. Right. And when you say they're relatively frequent, in terms of overall incidence of cerebral palsy, can you give me a sense of the frequency of that? Because it really struck me as being sort of a
Yeah, when I say they're relatively frequent, I also mentioned there's more than 300 genes that are being described in cerebral palsy. So they're frequent as in they're a couple percent, we think.
Right, okay.
They're not explaining 50% of cerebral palsies or anything like that. Right. But probably they're an under-recognised cause of cerebral palsy.
I've just had a question come through. From Nadim Shadiak from the University of Adelaide, it says, what's the therapeutic toolkit now or in the horizon for non-progressive disorders like CP? And what are industry talking about as being the silver bullet for spectrum-based disorders or disabilities? Thanks, Nadim, for an excellent question.
Yeah, I can't speak to what the industry are talking about. I mean, as far as the therapeutic toolkit for cerebral palsy goes, I guess really all we've got is those kind of early intervention physical therapies at the moment. If you can genetically diagnose some of these kids, some of them actually have disorders that you could treat. And if you treat them early, you can potentially completely remove their disability. That's not common, but if we don't do the testing, we don't know which kids would benefit from that. So there's a proportion of these kids who actually have a metabolic disorder, for example, where they could be treated. There's a lot of work going into diagnosing cerebral palsy earlier because it's actually not diagnosed technically until 4 years of age at the moment. If you wait until 4 years of age to start your therapy, your outcomes are not so great. So really, we're trying to tie in the genetic testing with that early diagnosis to give these kids the best possible treatment for their actual condition. Because really, cerebral palsy is just a group of disorders that are being lumped together by their clinical outcome. One cerebral palsy kid is not just like another. Indeed.
Claire, just as a means of follow-up, that you mentioned really earlier in the presentation that you are developing genetic panels to enable diagnosis. Can I get a sense of where they are in the development stage and how much do they detect all or 50% of kids with CP or?
Yeah, so what we've been focusing on is to generate a panel through PanelApp. And really that is just picking out the genes where there is sufficient support that they would be valid to use in a diagnostic setting.
Gotcha.
At the moment, I think we've got 73 genes in that panel. So it's certainly not a large number because you have to get enough families with the same genetic variant before you can use it in a diagnostic setting. But without that kind of gene panel, clinical diagnostic labs just can't do these kind of tests because they don't have the appropriate evidence to interpret a genetic test for cerebral palsy.
So of that, with that 70 plus gene panel, will you diagnose 50%, 10%, 90% of kids with CP? Can you give me a sense of?
It'd be a small proportion.
It would be a small portion,
so,
okay.
Yeah, it'd be quite a small proportion.
Yeah, no, look, thank you. My final question, and again, we're running rapidly out of time or have already done so, but I've got to ask, how many twins are you hoping to recruit as part of your study with the Hospital Research Foundation funded fellowship?
So our original aim was 50, but then COVID happened. At the moment, we're sitting just above 25, might be 26 or so. So we've done okay, but I'm certainly still looking to recruit more. And we do occasionally get inquiries about that. But yeah. Okay.
So any way of what we can transmit that, people watching this this webinar, tell all your friends that
tell all your friends, and if you think you know someone who might be interested, drop me an email and I'm happy to supply more information.
Fantastic. Look, thanks to both of our speakers this afternoon. Fascinating presentations. I wish I could ask more intelligent questions, but this is not my area of expertise, as you could probably tell, but I'm really grateful to really fascinating presentations. Before we close off, I'd like to remind people that these mini reviews are being recorded and are available from the ABMC website. So if you've missed anything during this live presentation, you can go back and watch it again, www.adelaidebiomed.com. Under the banner webinars, you'll be able to watch this in past webinars recordings. I encourage all of you online to remind your colleagues about these webcasts, and so that we can build awareness and make sure that people attend and see the great research that's occurring here in the Adelaide Biomed City and beyond. So without further ado, I'll just ask you to join us next week, same bat time and same bat channel. And thanks again to our presenters for this afternoon. Cheers.